Hello girls, I hope you're staying safe at home. To continue with the chapter Economic Importance of Bacteria and Fungi, today I'm going to talk about the role of bacteria in agriculture. Now, plants need nitrogen for making proteins. Why do plants need proteins? So that they can grow properly. But they cannot make use of the free nitrogen that is present in the air. I am talking about the nitrogen gas that is present in the atmosphere. They can obtain it only if the nitrogen gas turns into nitrates, because these nitrates dissolve in the water which is present in the soil, that is the soil water, and then it is absorbed by the plants and then it is made use by the plants in the plant body. Okay, so to convert this free nitrogen gas into nitrates so that it can dissolve in the soil water and can be absorbed by the plants, there is a special type of soil bacteria which is known as rhizobium. Okay, underlining it because it's a scientific name. So these type of soil bacteria or rhizobium is found living in small nodules, which is found in leguminous plants, in the roots of leguminous plants. Now, what are leguminous plants? Leguminous plants are plants which are rich in protein. Okay, so there are nodules. These are the nodules which is present in the roots of leguminous plants. So this bacteria rhizobium lives in the nodules. Okay, and what does it do? It picks up the free nitrogen from the atmosphere, and what is it converted into? It is converted into nitrates. So these nitrates are soluble in water which is present in the soil, and then the nitrates are used by the host plant, that is the leguminous plant in which these nodules are found. And of course the nitrates will remain in the soil. So later on, if other plants which do not have nodules in their roots are planted there, they can also make use of the nitrates that is already made in the soil. There are certain free living bacteria in the soil as well. Okay, to name some: Azotobacter. Please remember these names. Okay, and another one is Clostridium. These are bacteria which live freely in the soil. They do not need nodules to live in. It is only rhizobium which lives in the nodules. So they also use the free nitrogen in the soil and convert it into ammonia. Okay, with hydrogen, which in turn gets converted into amino acids and nitrates. And hence what happens? It will get dissolved in the soil water, which is again picked up by the plant. So it is beneficial to the plant. We are talking about free living bacteria in the soil. Okay. So next we move on to nitrifying bacteria. As you know that plants require proteins. Okay, and you know proteins are made from nitrogen. But the free nitrogen cannot be used by the plants directly because the nitrogen must first be converted into nitrates, which dissolves in the water and then it is absorbed by the plants. Okay, so apart from being fixed by certain bacteria, as I explained earlier, there is another method by which these nitrates are replenished in the soil by nitrifying bacteria. Replenished means it is made again. Okay, and the process is known as nitrification. Okay, so in the process of nitrification, what happens is some bacteria convert nitrogenous wastes of plants and animals, okay, and sometimes even the dead remains of plants and animals into ammonia first. The ammonia is then converted into ammonium compounds, which is turned into nitrites with the help of bacteria called Nitrosomonas. Nitrosomonas. So, Nitrosomonas is a bacteria which converts ammonium compounds into nitrites. Okay, so if I ask you, name the bacteria which converts ammonium compounds into nitrites, what will your answer be? Your answer will be Nitrosomonas. Okay, and after nitrites, the plants will require it in the form of nitrates. So the nitrites must be turned into nitrates, and this is done with the help of a bacteria which is known as Nitrobacter. So what if I ask you the name of a bacteria which turns nitrites into nitrates? What will your answer be? Nitrobacter. So each of these steps is brought about by a certain bacteria. Now next is denitrifying bacteria. Now when we talk about denitrifying bacteria, we are talking about bacteria which is present in the soil. And what does it do? It breaks down the soil nitrates, that is the nitrates present in the soil, and it changes it back into nitrogen gas, and which re-enters the atmosphere from where it came. So example of a denitrifying bacteria is Pseudomonas. What does Pseudomonas do? It breaks down the nitrates into nitrogen. So you can say the nitrogen cycle is complete. So what happens in the nitrogen cycle is nitrogen is taken up from the atmosphere. It is converted into nitrates in the soil. And again, after a certain period of time, it is reconverted back into nitrogen. That is one complete cycle. Okay, so you can see how atmospheric nitrogen is fixed in the soil and turned into nitrates by bacteria rhizobium, or industrial fixation in the industry where ammonia in the form of urea and other ammonium compounds, it is put in the soil. Okay, and even certain free living bacteria like azotobacter, clostridium, okay, it converts the free nitrogen into ammonia first, which then converts into nitrites and then into nitrates, and then it is taken up by the plants and it is used by the plants, and from plants it is used by the animals when animals consume plants directly or indirectly, and when plants die it decays. When animals die, it also decays, and when it decays again, it is reconverted into ammonia, further turns into nitrites, and again turns into nitrates. And denitrification bacteria which are present in the soil, remember Pseudomonas, it breaks down the nitrates and then it converts it back into atmospheric nitrogen, thus completing the nitrogen cycle. Moving on to the role of bacteria in industry, I already told you how certain bacteria are used in making medicines, antibiotics, and it is made on a large scale in the industries. Okay, another industrial use of bacteria is in taking out the flavors of tea. You all have heard about the flavors of tea. Darjeeling tea is famous throughout the world because it has beautiful flavor. Different types of flavors are produced by bacteria. So this is known as tea curing. Okay, and bacteria is also used in leather tanning. Now we have so many things made out of leather, like bags, shoes, jackets, so many other things you can think of, isn't it? Now the first step in making leather is taking out the skin of the animals and drying it. So when the skin of the animal is being dried, bacteria break down the soft perishable parts of the skin. Perishable means which can easily decay. Okay, and then the skin or the hide of the animal becomes hard, then only it will turn into leather. Okay, so leather tanning is also done by a certain type of bacteria. So this is the industrial use of bacteria. There are three roles of bacteria in industry. Number one, I already told you, in producing medicines. Number two is in taking out different flavors of tea. And number three is in leather tanning. Now you know, during summers when food gets spoiled, if you keep food for a few hours outside, if you do not keep it in the fridge, it starts stinking, isn't it? This food is spoiled by bacteria. It not only spoils food, it also spoils milk, fruits, vegetables. How? By the process of decay. That is also known as fermentation, and this you will see only during the summer. Okay, now this spoilage may sometimes be so much that it also causes food poisoning. You must have heard about food poisoning, isn't it? That is, if you happen to eat the spoiled food, you will get food poisoning and you have to be admitted to the hospital, constant diarrhea, vomiting, stomach ache. Okay, there is a serious kind of food poisoning which is known as botulism. Okay, it is a very serious food poisoning due to a special bacterium which is found specially in tinned food or any type of sealed food. Okay, the affected cans, you know, it gets, if now supposing this is the can, this is normal, but sometimes if you look at the affected can, it will get distended at the bottom with gas. Okay, and when you open it, this gas comes out with great pressure. So such this type of food should be thrown away. So preservation of food and making it free of diseases, germs is very, very necessary. So food preservation is very important. Okay, so food preservation can be brought about by different method. The first one is known as sterilization. Okay, sterilization means boiling or heating at high temperature. Okay, when you boil water, you are sterilizing it. You must have seen how bottles of little children are boiled. Why are we doing that? To kill any type of bacteria that may be there in the bottle. So boiling water kills all bacteria except their spores. So high temperatures, about 110 degree Celsius, at an increased pressure like in the pressure cooker, kills even the spores. The canning industry employs heating to kill bacteria before packing and sealing the food. That is, if you are taking canned food, remember that it has been sterilized. How is it sterilized? By heating at very high temperature in autoclaves. Okay, and then the food is put inside there and then it is sealed. Next is salting. You know, salting is a common method of preserving food, like fish, pickles. Okay, salted fish we have, pickles. So we can keep that for very long periods of time. When we mix the food with salt, what happens? The food does not get spoiled. That is, the bacteria will be killed if it comes in contact with the salt. Okay, so treatment with salt for preservation purposes is called again curing. Okay, now next we move on to dehydration. Or dehydration means you're taking out the water. You have dried fruits which does not spoil for a long time. Okay, so most of the microbes, that is the disease causing germs, they cannot grow without water. So if dehydration or drying of food is done, like grains, dried meat, dry fish, dry vegetables, so this type of food will never get spoiled. We also have dried milk powder, which is another very good example. Okay, next is irradiation, means radioactive radiation has been done to sterilize certain food without themselves becoming radioactive. But of course this practice is not much used. Okay, so ultraviolet light is found useful not only in sterilizing the air in schools, hospitals, and factories, but also it kills mold spores in bakery. Okay, next is pasteurization. Pasteurization is a technique of partial sterilization, specially done only to milk. If you look at some milk packets, you'll see it is written outside in the packet, pasteurized milk. So how is pasteurization done? The milk is heated up to 60 degrees Celsius for about 30 minutes. Okay, and then it is put in the freezer, chilled quickly. Okay, so when this is done, you will get pasteurized milk. Pasteurization kills majority of the bacteria that are present in the milk, even the disease causing one. Okay, but remember, pasteurization does not completely sterilize milk. Okay, so bacteria which spoils the milk may also be present in the milk, but of course we have to keep the milk cool enough so that these bacteria do not multiply and makes the milk sour. Okay, next is of course refrigeration. Cold temperature, like certain disease causing bacteria and molds do not grow and multiply at a temperature below 0 degrees Celsius. So chilling of milk and keeping vegetables and fruits in cold storage are popular methods of preserving them. We also have frozen fish or frozen vegetables, and it can be kept fresh for a long time in the refrigerator. Okay, and of course the refrigerator that we use at home cools at from 0 degree to -5 degrees Celsius. But of course we cannot keep food for a long time in the domestic refrigerator. Okay, deep freezing cools at -20 degree Celsius to -30 degrees Celsius for long-term storage. That especially in shops and all, they use deep freezers. Okay, now next is strong concentration and chemicals, like jams, pickles do not get spoiled because jams have a strong concentration of sugar, and pickles have a strong concentration of salt. Okay, and in some cases preservatives like sodium benzoate are also added to tinned foods and squashes. So moving on to bacterial diseases in plants and animals. Okay, so in plants there are two diseases that you have to learn, that is black rot, you'll find in mustard and cauliflower, and next is bacterial blight, found in cowpea. Okay, now moving on to bacterial diseases in animals. Okay, you have anthrax, especially in cattle, cows, ox, okay, buffaloes. You will see that in anthrax what happens? Swelling on body and reduced milk yield. Tuberculosis, you know that the lungs are affected and there is a dry husky cough. Okay, now next is In humans, there are so many different types of bacterial diseases: whooping cough, cholera, tuberculosis, diphtheria, typhoid, pneumonia, tetanus. All these can be cured with the help of antibiotics. Okay, now next is a term which is known as bioweapons. Okay, now bioweapons are nothing but these are germ bombs, which when exploded, what does it spread? Disease, killing millions of people throughout the world. Okay, and one such bioweapon is anthrax bacteria, which may be sent to various countries. Okay, you don't need guns to fight. What do you need? Germs. You send the germs on a large scale, and the many people will get sick. It takes an epidemic shape, and then there will be lots of death generation after generation. So with this, we come to the end of part 1 of the chapter Economic Importance of Bacteria and Fungi. Stay safe, children. God bless you.